Good morning, everyone. I'm very glad to be here to present one of my favorite projects in my lab to you. And just by looking at the slides, you probably not see any nanoscale objects. Actually, I think that's a future for nanotechnology. The nanotechnology should be, in the future, already integrated with many things, many portable sensors, or even in our bodies. And we're not supposed to see nano material or device because they're so small. But our research goal should always be to translate the research of nanotechnology in the laboratory into our everyday life. Okay, so this is exactly what we do in our lab. We're trying to translate the nanotechnology into your everyday life. So one particular technology we're trying to develop is to integrate nanoscale sensor materials with mobile electronics like cell phones or mobile phones, we call it, or smartphones, which is most people use right now. And to actually give the smartphone user, which is everyone in this room, I believe, the power of detection or diagnostics at the molecular precision. And we cannot do that at this moment yet, but my vision is in 3 or 5 years, we can translate the technology in nanoscale from the laboratory here into your phone. Okay. So just for looking at the pictures, you can see, I can give a very brief introduction of what this is about. This is basically 2 projects I'm going to present today. One is what we call mobile phone water sensor technology based on a nanoscale electrochemical sensor. Another one is our recent development of integrating nanoplasmonic sensor onto cell phones so you can do surface plasma resonance imaging on phones instead of using very expensive or bulky equipment. And our target, of course, is always on health and environmental health. This is the Cancer Biotechnology Institute, right? So I want to start my talk from the cancers. Okay, when we do cancer research, this one. First of all, this is just another showcase showing what we do in a laboratory. You don't see this in your everyday life, but our goal is to have all of this showing on the slides to integrate with some sensors on your smartphone someday so you can utilize the powerful functions of those nanostructures and nanomaterials for biological applications. Okay, and here is basically semiconductor nanomaterial we made in the lab, and this is microfluidics, and this is integrations of cell imaging with nanostructures to enhance the sensitivities or improve the spatial resolutions. And also you can see that we're working at interface. Well, we heard a talk from Professor Pan early this morning that he's talking about interface between biology and medicine and engineering. Here we're working the same line. We're trying to work in nanoscales to integrate molecules or cells with nanomaterial or device to create new functions. Okay, but I want to start my real talk to just by looking at the slides. When we do cancer research, our ultimate goal should always be we want to develop a way to stop cancer, to let everybody in this world to live in a cancer-free life, right? Of course, it's still a dream, but everybody in this field is pursuing that dream now. And there's many people working in different aspects of cancer prevention, diagnostics, treatment, and prognostics, right? So let's start from the preventions. So we know, actually, we are what we eat and what we drink. So we can effectively actually prevent or deter the development of cancer by drinking clean water or eat better food or clean food. And in fact, many people get cancers partially because in some point of their life, they start drinking dirty water or food, or eat dirty food that contains carcinogens. Right? We can find many carcinogens in those contaminated water or food, but we actually lack some powers to really identify them because they're usually very small and they're usually are not colorless, and there's no smells. So if you see a clean water, it looks like very, very clean, but actually it could contain some levels of carcinogen in there. So our goal is to basically have some nanosensors integrated with smartphones to allow people to easily identify carcinogens or other invisible chemicals that we cannot detect before. So one of the key issues in the United States is water safety as well as related to food safety and health. And while it may be a very surprise to you, even though this is probably a very rich country, very highly developed country, but we still have 15% of people in this country don't get their water from the tap. They get their water from their private groundwater wells. 15% of this country getting their water not from the tap, but from the private well, which are not subject to EPA regulations that can be contaminated by all kinds of sources. Okay, and give you one example. Arsenic is a well-known carcinogen, and the arsenic in drinking water resulting in very serious toxicity as well as cancers. And this happens in India or in Bangladesh or other Southeast Asia country, or of course is also happening nowadays in the United States, especially in Central Valley, California. Okay, and there's communities that actually suffer from drinking water with a high level of arsenic in California. And in this world, every 20 seconds, a child die because of drinking dirty waters. So it totally, about 4 million people die because they don't have access to clean water. So, give you another story. This is a really, really famous story. For those of you who like to watch movie, you probably watched the movie Aaron Brockovich, right, played by Julia Roberts. And this is really a very famous lawsuit in the history of the United States. And the settlement amount is about over 300 million US dollars. This is because the actual discovery of a residence in a small town in California, many of them, a high percentage of the residents in that small town have cancers. And people start to trace what's the reason for that, and then they actually eventually find out in their drinking water there's a high level of 6-valent or hexavalent chromium because of the leaching of the gas or oil pipe from the Pacific Gas and the electric corporations. So there's a very, very long haul lawsuit, and eventually the residents win and have the settlement in over $300 million. So that actually tells a story. Back then, in 1990s, about 20 years ago, people really don't have the power, even though they're drinking water every day. It looks very, very pure, very clean water, but they have no power, no capability to really find out what's in the water. Not even mentioning to detect water quantitatively and tell the information to their neighbors or their friends. Okay? But now, 20 years later after that instance, we want to create technology to enable people, to empower people, that they know what exactly they're drinking and what they're eating. And if there's a high level or excess amount of carcinogen or other chemicals, they can not only alert themselves, but also can send information to their friends. All right, so in this area, okay, that was a story in California again. This is a story really happening in this location, in the Midwest areas. Because we have a very heavy agricultural activity over here, so farmers here, they always over-apply fertilizers or pesticides up here. So you can expect, especially after heavy rains, you can expect a high level of nitrate or other chemicals you can find in fertilizers existing in the groundwater, or sometimes even in the local residents' private well ground well. So especially for nitrate, if that residents happen to have a young baby at home, they have to feed the baby using baby formula or milk powders. They have to mix that ground well water with the baby formula. And then if you have excess amount of nitrate in that water, the baby will turn to blue baby. Why? Because the nitrate will compete with oxygen, the bind with hemoglobin in the red blood cells, and then to deprive the oxygen from the baby's systems. This is actually sometimes is very fatal. So this is even more serious problem than cancer. Cancer, you can wait for 10 or 20 years, but this you cannot wait. It's happening instantaneously once you drink that kind of water. Okay, and so both WHO and EPA have the very strict regulations of how much nitrate you should have in your drinking water. It really is supposed to be much lower than this concentrations. But of course, if we trace the water, because all the waters are connected underground, so the water over here, the contaminated water over here, eventually will end up into Gulf of Mexico, because all the groundwater eventually will go into Mississippi River, right? And then that actually flows down to Gulf of Mexico. So that's why people, every year, especially every summer, people find a very high level of nitrate along the coast of Gulf of Mexico. That caused not only the problem to people, but also caused problem to fish living over there, and a lot of fish dies because of that reason. Because you have, the nitrate is also toxic to them, as well as it actually deprive oxygen in the water, so the fish will die. All right, well, this is another story which happened to my students. This is also one of the motivations why we want to invent the technology. One of my students actually took a trip in 2011, is engineering without water trip to Guatemala. And their main missions to basically help the local residents, usually very, very poor communities, to evaluate their drinking water qualities. And so back then, they only bring very rudimental tools over there, so they can only get very limited information on the water quality. What they have to do is they have to sample the water from different locations of the village and take it back to the United States and send to specialized lab for more quantitative analysis, right? So they cannot tell the villager the information right away. They have to wait. They have to take sample back. My students have the motivation or have ideas. If we have a much more powerful tools, especially can couple with the smartphones, and to get information at that point, that will be really much more helpful than doing in this way. So we start to identify what's the technology gap we can fill in, right? So now the technology gap is you can find a very low cost but very crappy so-called quantitative tools or assays which only shows color, tells you yes or no or high or low. There's no quantitative information at all. And on the other hand, you can find very expensive, very bulky, facilities in the Department of Chemistry over here. You can find HPLC, LCMS to do a very quantitative analysis of chemicals or molecules in liquid. But there's a gap between. So can we do really low cost, device or sensors, but with a similar, if not better, powers as these quantitative expensive tools? So that's a technology gap we want to fill. Okay, this definitely requires some innovations and even revolutionary innovations because the traditional technology does not need to do that. So you have to really use nanotechnology to have a similar sensitivity or power, but with much smaller size and cost. Okay, so just use nitrate detection as example. As you can see from this picture, if you see a water like this, you probably will not drink it because it's already dirty enough visually, right? But if you see a water like this, you don't know which bottle to drink if I don't label them, okay? But I label them here. One is the actual water poured from the bottled water. The other one is the same water but spiked with high concentration of nitrate. If you drink this, your skin probably will turn to blue. But you cannot tell which one actually are drinkable water just looking at it, because there's no color, no scent, no smell. There's no way to actually identify them. So what we're trying to do is invent a socially connected water nanosensors on mobile phone for both environmental and mobile health applications. Okay, and our ultimate goal is to really empower every one of you and all the smartphone users the ability to sense invisible water contaminants and biofluids on phone. Okay. And to broadcast the citizen participatory water sensing information. I think that's a really critical piece of a smartphone technology, that you not only get information yourself, you can broadcast information, you can share information, on social network. And of course, we want eventually, of course, to connect your personal health data to your healthcare providers more securely in that case. All right, so this is an overview of our technology solutions. As I said, you will not see a nanoparticle here because our goal is really integrate nanoparticle in a complete system. You don't see them, so you can use them. There's always no way to use a nanoparticle. They're too small. You cannot even pick them by your finger. So you have to integrate them monolithically and or organically with other systems. In this case, we are leveraging the nanosensors, mobile phone, cloud computing, and social media, all the powers together to complete a platform technology, allow people to detect the invisible contaminants, including carcinogens or other toxic chemicals, and then use phone as a computer, basically, to control the sensor, to digitize information, to send information wirelessly, and then couple with social media to broadcast information, and then couple with cloud computing for further data analysis or for geographical information system services. Okay. So eventually, we want everybody not only become someone that can protect themselves, but they can become an environmental scientist to help the whole community. Okay. Now this is the sensor configurations. Just by looking at this, again, you cannot see nanostructure, but I actually show some nanostructure here. It means that we do have nanostructure or nanotechnology here, because without nanotechnology, you just don't have the sensitivity, don't have the power to detect the low concentration of chemicals in water. Okay, you have to use this to improve your sensitivities. But you also have, we are also working on, we're not just, stopping at this step, which is, I think, is one of the biggest roadblock in the nanotechnology field. Many people stop at this step. They never really go beyond this step to integrate this nanotechnology with the complete system. But this is what we do. We integrated the actual nanosensors into microscale, then eventually to mesoscale, and then have a complete package that can be coupled in the phone. So this is a very simple design. Basically, we have 3 electrodes, microelectrochemical sensors. There's nothing fancy about this, but the typical electrochemical sensors has very limited or modest sensitivities. To improve the sensitivity so that we can detect, let's say, 10 milligram per milliliters of nitrate in water, we have to make nanostructures on the electrode surface to, one thing is to increase the surface area to increase the electrochemical redox potentials or current. On the other hand, by using nanoparticle on the surface, we can really increase the electrochemical turnover rate of redox molecule that you can find in the liquids. So just by taking this power of nano structure and in nanoparticles, we can basically improve the sensitivity of electrochemical sensing for about 100 to 1,000 times. Okay. And of course, then you have to integrate that with microfluidics and to have somewhat automatic liquid sample powers, and then integrate with electronics and to the smartphones eventually. So this is an overview of the whole platform. Of course, we don't show nanotechnology here, but we do have nanostructures on the electrode surface, but this is a microscale. And then we integrate that with a circuit board and then with the phone. Here we are leveraging the DA and AD on the phone electronics and to reduce the cost and also improve the power. And then, of course, this phone has to be covered with, it has to be coupled with the social media and then eventually to the private internet cloud. All right, this is our pattern. I'll skip this part. Just give a very brief introduction of what is electrochemical sensing. Electrochemical sensing is a very straightforward concept. Whenever you have ions in water, those ions potentially can exchange electrons with electrodes. You can either donate electrons to that ions or accept electrons from the ions. And that, that so-called reduction and oxidation potential and the current is dependent on the concentration of the ions and ion species. Okay, so just by looking at that, the redox potential and the current, we can tell which ions we're detecting and what's the concentration of the ions. So we categorize our device and compare with a macro scale commercial electrochemical sensors, and we find out we only need to use 70 microliter. Of course, this volume can be even lower. We only need to use 70 microliter of the liquid volume in our detections, and our limit of detection is about 0.4 micromolar, which is about 25 parts per billion, which is much better sensitivity than a macro scale commercial sensors. Of course, we, our students, once they make this sensor systems, they have to go out to test them. So what they do is they actually travel to different part of the country, including LA, San Diego, Chicago, Urbana, of course, and Muhammad, which is about 20 minutes drive from here. And they collect water from different places, including stream water, Crystal Lake, which is local lake, and Lake of Woods, it's a lake in Muhammad, and Los Angeles and San Diego and Chicago, Michigan, lake waters. And they take that water samples and use multiple detectors or multiple sensors, including ours. So ours is this red color ones. We call that electrochemical microsensors. And then we compare with many other sensors you can find in the market, including this yellow one. This yellow one is cadmium reduction assay, which is considered by most chemists as a gold standard in this nitrate detections. So we particularly care about the comparison of our data with this yellow data. So if you look at the red and the yellow data, they're pretty consistent, except a few samples from Michigan Lake. And then later we find out it's because in those waters, you not only have a nitrate, but you also have a low trace of nitrite. But our sensor cannot detect nitrite, but this cadmium reduction can detect nitrite. That's why the yellow data is always slightly higher than our data. But just from this data, from this graph, you can see that just don't trust blindly when you're buying a sensor from the market, right? Because they don't provide very consistent measurement data. This is all sensor you can buy directly off the shelf, but they don't give you the same answers. Okay, so the operation we're trying to make it very simple, because I think that's the future of nanotechnology. Because nanotechnology, we're dealing with the materials of 100 nanometer or smaller, but we are people of 100 pounds, 6 feet, right? So you have to really make a operational system that includes nanotechnology in there for the detections. So we trying to integrate into a very complete system, so you can do a simple water sampling just by utilizing the capillary force in the microfluidics on the sensors to do the detections. Of course, we also have to develop our own software, including this mobile phone app to do a sensing and control and data acquisition, data processing, and communications. So that's as I said, this is our first step to trying to really give the people the power to see whether the water they're drinking, the food they're eating are clean enough. But eventually, we can use a similar technology, because this is basically liquid sensing technology. We can use similar technology to detect those invisible chemicals or biomolecules in body fluids, such as blood or urines or tears or sweat samples. Right. So this is our very recent work to show that we want to do not only blood glucose detection, but also at the same time getting the readings of a few other molecules you can find in the blood, in the 1 drop of blood. And this, of course, the final goal of this is also many people working in the mobile health field. You want to have nanosensor integrated with the phone, and then have information on the phone, gathered on the phone, and sent to your doctor through this wireless network. And then this is our very recent data. We show our glucose sensing sensitivity and accuracy is very comparable to the off-the-shelf glucometer sensors. Okay, so I forgot to tell you that, I told you about 4 million people die every year because of drinking dirty water, but most of them actually die not because drinking water contains nitrate, okay? Most people die because their drinking water contains bacteria. So this is actually even more important for us to do is to develop a cell phone bacteria sensors in water, for water samples than nitrate samples. So we have published data about 2 months ago on sensor and actuator B to show that we can have integrated package containing microfluidics and some nanopore structures on this serpentine electrodes array to do a so-called impedance spectroscopic detection of bacteria, including E. coli and Salmonella. And we can quantify them. We can also identify the species of the bacteria. Of course, at this moment, we cannot detect the subtypes of the bacteria. For example, if you give me E. coli K1 strain, or you give me E. coli 0157 strain, I probably cannot tell the difference by using the sensor. But if you give me Salmonella and E. coli, I can tell you which one is which, and what's the concentration of each. So this is our, we're trying to develop a really portable, a very compact sensor by integrating nanotechnology and microtechnology onto the sensor to couple with the smartphone for bacteria sensing. Yeah, this is actually showing some details. We have microelectrodes in this so-called interdigitated electrode format, so you can increase the capacitance when you do the measurement. And then we have a sub-micron pores in between those electrodes to allow the sub-micron microbial to pass through, but to leave other bigger microscale particle away from that detection chambers. So our ultimate goal, of course, this is our checklist for both drinking water detection and biofluid detections. So we are now already accomplished nitrate, phosphate, arsenic sensing, and we're set to actually develop more functions for detecting other contaminants in water. For body fluids, we already demonstrated we can do glucose, cholesterol, and a few other items, and we're, of course, now trying to move to a more challenging biomarker detection, especially cancer biomarkers. So eventually what we want to do is, in one drop of blood, can we detect a low trace of cancer biomarker and quantify them? Let's say, can we detect CA125, or can we detect PSA from a urine sample of a prostate cancer patient? That's what we want to do by using this sensor. All right, so this is a so-called GUI, or graphical user interface of the phone app for what we call crowd sensing. You're not only doing the sensing yourself, you're coupled with crowdsourcing of the social media to send information, broadcast information. And what we do, we actually work with the Twitter together to couple our information feed to a Twitter account. So everyone subscribed to the Twitter account will get instantaneous information, not only from one person, but from the whole community using this sensor. But this is what we want. What we want to do eventually is by working with the government together. We're trying to integrate the so-called citizen nanosensor network data with government immobile sensing network. For example, this is an integration of data from USGS, which is U.S. Geological Survey. It's a government sensor network data with our own data, and then coupled with the local weather stations, local sensor station data together to give people this rich information on Google or Bing Map. Okay, so this is ultimate goal for this, and this really serves as a very rich environmental health sensing big data map to help everybody. Okay, so this is a visualization of the sensing data on the phone, and so for the end of this project introductions, I want to acknowledge our collaborators and some of our founding agents, including we're actually collaborating with national grade reverse research education centers. They're located somewhere very close to St. Louis, and they have a very nice K-12 educational program. It's called a River Watch. So every summer they have a local high school students from different schools from St. Louis or other part of Southern Illinois, and then we're trying to really make those low-cost sensors and give one to each of the high school students, so they can use their own smartphones for detecting water quality in their local communities. And then that information that can be all gathered into a common cloud, and especially that information will be very valuable for the National Grid Reverse Research and Educational Center for further studies. Because once you have 100, 1,000, or even 1 million people start to contributing data from different part of the country, and that data is really valuable, I can tell you. Yeah. So, of course, we got funding from many different places, including Nokia, Vodafone Foundations, NSF, NCSA, and NCAA. This is this main funding source for this project. All right, so I mainly talk about the water sensing, which is more like a preventive medicine. We want to prevent you from drinking dirty water, so you don't want to drink any carcinogen water, and that may stop the cancer at the first step. But let's say for other reasons, carcinogen is one of the reasons for cancer development. But there's also other reasons for cancer, for example, genetically. So even when you drink very clean water for your whole life, you may still develop cancer because of your genetics. Right? So in that case, we really want to develop a nanosensors for cancer diagnostics, or for people already undergoing the treatment. We wanted to do cancer treatment evaluations or prognostics. Let's say we want to predict whether this treatment is effective or not, or how many years this patient can live, right? So the ultimate goal of this community was so-called mobile health community, which is considered by many people as a healthcare of the future, is eventually you want to have really integrated sensor technology, potentially on your smartphone or on your iPad. So then for the most of the time, you don't have to go to hospital or visit your physicians, and you can only just do very simple detection at home on your smartphone and get all the molecular level informations, and that information that can be shared through the wireless network securely with your doctors and your healthcare providers. And people already start doing that, including those big corporations like Microsoft, Qualcomm, Google's. They've been doing this for already several years. They're trying to link the health sensor information with information technology or internet to really share information with your doctor or with some medical researchers. And there's in terms of individualized molecular medicine, there's already many early adopters. One of a really good story I want to talk about again, actually I talked about this many times already, is the story of 23andMe. If any one of you already used their service, probably you know this company very well. This is a company started by the founder of Google, Sergey Brin, and his ex-wife, Anne. While this company actually is set to give everybody the service to analyze their own genes by sending a kit to your home. Of course, you have to pay about $100, and then they send a kit to your home. You then donate some saliva and send it back to Google or to 23andMe. In probably 1 or 2 months, they can tell you whether you have type 2 diabetes, or you have the genes susceptible for cancer or susceptible for Alzheimer's disease, right? In fact, Sergey Brin himself used the service of 23andMe and discovered that both he and his mother possess a mutation of gene that is associated with Parkinson's disease. In fact, his mother just developed a symptom of Parkinson, and he's himself was predicted to have this symptom in probably 15 to 20 years. Okay, so this is the power of so-called molecular medicine or individualized molecular medicine. But of course, still now the current technology only allows you to do this ways, which means you have to donate your biosamples, your biofluid samples, like saliva, and send to the company like this. But in the future, what people really want is to give the power completely to you. Let's say you got a sensor already integrated on your phone. You don't have to send anything. You just click on the sensors, and in just a few minutes or in 1 hour, you can read from the phone what kind of genes you have, whether you have cancer, subtractive gene, or you have Alzheimer's gene. So that's the future for this so-called mobile health. And then, in terms of genome, in terms of genetic study or genome sequence technology, particularly, we have seen a very rapid change or a very rapid development in the past 15 years. Starting from 2000, Affymetrix starting to have their first so-called DNA microarray technology to really do genotyping for everyone. And then over years, we see the company like Illumina, like Pacific Biosciences, Ion Torrent now is part of a Life Technology, to really offer smaller, but more and more powerful nanotechnology to sequence your whole genome, right? Sequence the whole genome. So now this is really what you need. This is how big of a device you really need to sequence your entire genome, the 3 billion base pairs of your body. And one of the good stories that this company, Ion Torrent, they actually utilize something called ion selective field effect transistor technology, which is not too much different from the technology Dr. Gordon Moore, who is the founder of Intel, have tried to develop over the past 40 years in Intel's. Right? The Intel technology is basically based on MOSFET or CMOS technology. But people now start using a technology very similar to CMOS technology to sequence the entire genome of Dr. Gordon Moore. This happens in about 3 years ago. Okay. So now the actual nanosensor is really becoming more and more powerful. You can even use that to sequence the whole genome. And there's even more crazy stuff nowadays. There's a company called Nanopores from UK. They're pledged to really develop a desktop-based or even laptop-based USB whole genome sequencer, so you can use your own laptop to sequence your whole genome. This is really crazy. All right, but there's still there's some missing links in this mobile health world. On one hand, you see a rapid development of this bio-nanotechnology. On the other hand, you see even better development of information technology. We all enjoy our lives by taking advantage of the information technologies. But there's a missing link between these two to link this together, so then we can really leverage the power of both. Right? There are some people who have tried to do that. For example, Abbott, which is a company located in Chicago, they have a product called iStat, which is a very nice product, but it's too expensive for most people. You have to spend over $10,000 for each one. This very commonly used in the bedside diagnostics in the ICU in a hospital. Okay. But our solution, we're trying to really utilizing nanotechnology to make a very powerful molecular diagnostic tools much smaller and much cheaper and much affordable and accessible to most people. One example I want to give you is, so this is one of the projects in our labs. We're trying to really shrink down the SPR technology. For those of you who knows SPR, you know that that's a technology that has been used by many pharmaceutical companies or even the hospital to do molecular screenings or to study interaction of drugs or proteins or study the binding of DNA with proteins. The concept is quite straightforward. You basically have just a thin layer of metal like gold or silver, and then at some specific angle, you can excite the surface plasma resonance wave, which is a collective free electron wave in this metal film, okay? And that it is angle dependent. That's why if you sweep the angle, you can always find different reflectivity. So then by recording this reflectivity versus instant angle spectrum, you can find a specific peak that corresponding to the surrounding refractive index, optical index of the material. So let's say you have a molecule absorption, you have a DNA-protein interactions on the surface, then this particular peak position will shift. By detecting the peak shift, then you will know then you have molecule interaction on the surface. Yeah, so this is the concept illustration for the SPR. Then you have, in any metals, metal is a conductor because it has a lot of free electrons. But if you excite these free electrons using some certain frequency of light, then you can excite the wave of electrons propagating along the surface. And that wave frequency is determined by the dielectric constant of the surrounding media. If you have a molecule bound to the surface, then obviously this wave frequency will change. That's physics behind this. So conventionally, what people do is they have to use a very powerful, very big machine, and of course very expensive at the same time. One of the leading products is from GE Healthcare. It's called the BeerCore SPR sensors, and that actually allows you to do many different things, including protein-protein interactions to study, or even cancer drug molecule screenings, or immunoassays development. Okay, but the problem is you need such a big machine, and you need to have a really deep pocket to buy this, right? But our goal is, can we really shrink this technology on your palm size, or even integrate with your cell phones, so we can enjoy the power, but at the same time make that really affordable to everyone? Okay. So we have to really do the revolutionary research, not just incremental research, to completely change this actual technology to make that accessible to anybody, to everyone. So here it shows that we use what we call a nano copper ray technology, and to replace that bulky and expensive machine. In this case, we just use a plastic film to make a nano copper ray coated with gold or silver nanoparticles. And that kind of film is very special because it can change color, which can be identified by your naked eye even. It can change the color when you have a molecule binding on the surface. So instead of using a very expensive equipment like this, you can only use your naked eye, or just use your the color camera on your smartphone to take a color picture and identify what's molecule binding on the surface. Okay, so our goal is to try to develop a mobile phone SPR nanosensor. The cost should be lower than $100, and the weight is actually very, very low. And people have been doing this nanoparticle or nanoplasmonic thing for a long time, but our contribution is we integrate everything onto a really functional device. And we got our inspiration from this Roman technology. This Roman technology was invented about 400 AD, so 1600 years ago, okay, by mighty Romans. And they're really smart back then. They know how to mix a nanoparticle of silver or gold with the glass. So basically, they impregnated a lot of the silver particle or gold particles on the sidewall of this glass wine cup. That is why this wine cup exhibit red and green color depending on the illumination conditions. But we actually learned from that, and we also use our nano fabrication facilities here to shrink the size of this cup for about 10 million times. But that would be too small for one cup, so we make about 1 billionth of such a nanoscale cup into an array and put them in a highly water fashion onto this plastic film. And that is why this film exhibits a very similar optical property as this macro scale cup made about 1600 years ago. That's why we named this nanotechnology, or this nanosensor, as a nanoelectronic copper ray. Okay. All right, so this is a nanoscale details of that. You need to create a cup structure which has a bigger opening but narrower bottom. On the sidewall of this cup, you have to coat nanoparticle, which is the crucial part of this fabrication. Without that, you will not have this special property of the sensor. And then here shows what what I talk about. If you put different things on the surface, it will show different color. For example, if you put oil here is red. This is a ethanol and a salt solutions. They all exhibit different colors depending on their optical properties. In this case, you don't have to use any equipment. If the scale is big enough, just use your naked eye. You can see the color directly. If this is too small, of course, you can use a magnifying lens to see that. Okay. So we use this technology to identify different liquids just by looking at their color difference. Okay. And this paper was published as a cover article in the first issue of Advanced Optical Materials, which is optical section of Advanced Materials. And then this story is picked up by many magazines, including Wired Magazine and even Forbes. It's because people think, of course, you got inspiration from Roman technology, that's one thing, but also our goal is to replace a $100,000 technology by a $100 technology, and in the same power. So I think that's the future of nanotechnology. You can really revolutionize things to make some things which is not accessible to everyone become accessible. And then we show that we can do a DNA microarray just by looking at the color difference, right? Before people have to use fluorescence tag to label DNA, and have used something called fluorescence scanner to do a DNA microarray imaging. But now we don't have to use those things. We can just simply immobilize DNA on the surface to do DNA hybridization and detect double-strand DNA from the single-strand DNAs. Because after hybridization, the color, the actual visible color will change. This can be simply picked up. If the spot is big enough, you can see it by your naked eye. If it's small, you can use a magnifying lens plus your color camera to see this. Okay. Of course, to actually operate a biofluids or protein solution or DNA solution, you have to integrate that with the microfluidics. This is exactly what we do. So this is a really good picture showing you the power of this device. We have 10 parallel channels here, and in this 10 parallel channels, we inject 10 different liquids. Without this device, you basically cannot see any color difference because those liquids are completely colorless. There's no color. That's why they all look transparent outside this region. But once they enter this nanoscale region, you actually can see different colors depending on their optical properties. Okay. So of course, with the power of image analysis, you can do a more quantitative analysis, study of this liquid property, including the mixture proportion or concentration of each liquids in those samples. And here we show that we can use this colorimetric things to measure the real-time supertavidin and biotin binding on our device surface. We can record this video over some time and then calculate this spectral wavelengths positions and correlate with the time and show that we can do a kinetic studies of a protein-protein binding by using this very simple low-cost device. Of course, we can also study the contact printing of biomolecule on the surface. This is showing we use microcontact stamp to contact polylysine, a monolayer of polylysine on the surface of our device. We show that we can pick up those molecules just by looking at the color difference. Of course, besides the molecule, you can put the cells on the surface. We grow cells on our device surface and use color imaging to identify the cell membrane properties. Okay. Because of the time, I probably need to go a little bit faster. So what we do here is, of course, we are taking color image, and we use our algorithm to translate RGB value in our color image into a spectra. And then we can extrapolate a spectra peak positions and use that to do more quantitative analysis. And what we can also do is we can we can use a single color LED as our light source and measure the intensity change on this sensor. This actually give us even better reliability or better linearity. This shows that we can do very linear categorization using this method. All right, so this is the actual ultimate goal, and we're already halfway through that things. So what we're trying to build is a cell phone-based DNA microarray. So we want to do genetic or something called genotyping on the cell phone. Use a cell phone color cameras, but in that case, you also have to use our SPR sensor and a very simple single color LED light source. And this is our first generation of the setup, which is a very simple optical tower. You have an LED on the back, at the bottom, and you have, and we have a sensor in the middle, and use a iPhone camera to take the image. Of course, we have iPhone app to do a image analysis and to quantify the RGB value and then translate that into a spectral value. All right, so with this, I want to summarize my talk. So I've been talking about primarily how do we integrate nanotechnology into a complete system for biosensing, either sensing of water or sensing of body fluids, okay? And our ultimate goal, again, is to do a better environment and health sensing. And in the end, I want to acknowledge all the funding really contributes to this project. We really appreciate the funding from White House and the US DOE because they are the initial funding get us to start the project. And then once we start the project, we get more funding from Vodafone Foundation and Nokia. Of course, Nokia is one part of Microsoft. And this is really crucial for us to have everything here. Thank you very much.
So, when you mentioned very briefly that you could differentiate between different bacterial strains, like E. coli versus Staph aureus or whatever.
Yeah.
What was that based on the biophysical characteristics of the bacterial shape, or you conjugate antibodies? Or?
No, I think for different species like that, you don't have to use antibody to to identify them. You can just simply use their something called dietary property, depending on their size and shape and cell membrane permeability to differentiate them. Yeah.
Any other questions, comments? Yeah, let's go there and we'll come back to you.
I just had a question about the SPR sensors and how sensitive they were compared to traditional ways of monitoring those types of things.
Yeah, that's a very good question. So when we do SPR sensors, usually we use units of nanometer per RU, which means how many nanometers wavelength shift you can observe if you have if you have change of one refract index unit. Okay, so I want to just go back to the slides, compare this to the actual typical thin film-based SPR sensor has a sensitivity about 200 nanometer per RU. Okay, but ours, depending on the sample, depending on the size of molecule, especially for small molecule, we have a higher sensitivity. So our high sensitivity, it's about 500 to 600 nanometer per RU. So it's 2 or 3 times higher than the conventional technology. Yeah.
This is you're using a camera to detect the wavelength shift depending on the color unit?
Yeah, so we do something called translation algorithm. So what we do here is we first to categorize the spectrum of our light source, okay, and then correlate that with the RGB value we can detect on the smartphones. And of course, at the beginning, you have to use some color standard to establish that kind of a calibration curve. So once you have a color image from a sample with a known RGB value, you can then translate that RGB back into spectra. and then using the spectral peak for more sensitive sensing. Okay, and for we have other way of doing it is just use a single color LED. In this case, we're not measuring RGB, but we're measuring intensity. In that case, it's more straightforward. You just look at the intensity change, then to do the biosensing. Okay. Right.
Yes, you have a question?
So you're able to do this with like clinical samples like blood, urine, or you're not there yet?
For urine, yes, we're doing it. For blood, it's more complex. You still need to use some filter. First, you have to block all the cells, and then even for plasma, sometimes it's turbid. Okay, so yes, there is some challenge in the sample preparation step, but for urine sample or sweat, it's much easier. We can do the right away. Yeah.
Yes.
So are these sensors recyclable? Or?
Those sensors definitely recyclable, but we make it cheap enough so for biosensing field, they are almost can be treated as disposable. Yeah.
In the case of environmental smart sensor, is it for the individual or for the researcher?
Well, for individuals, of course, you need to have a sensor with ability detecting not just one, but maybe multiple contaminants. Then, at that point, it'll become much more valuable to individuals. So at this point, our water sensors particularly are primarily used for research purposes. And we have a lot of collaborators from the environmental science field. And they're interesting by using our sensor to study the water qualities in our local water systems, which is not difficult. Which is very difficult to do because always you have a very big land, but not enough manpower to do that. Yeah.
Yes.
Question.
So how exactly do you ensure the selectivity of materials for the
Yeah. So our selectivity for the electrochemical sensor comes from, as I said, from the reduction and oxidation potential and current. That is ion species dependent. For that case, we don't have to functionalize our electrodes. But for SPR case, yes, you still need to functionalize the surface with either targeted DNA or antibody or aptamer to identify or to be specifically targeted to those molecules you want to detect.
So what I'm assuming is though, if it's in the environment, there's no multiple species of water. So how do you do?
Yeah, so let's say you have arsenic ions or nitrate ions together in your water samples. And they will have different reduction and oxidation potential, which you can tell by looking at the position of, here. So this is our raw data. Typically, we scan the voltage from negative to positive and then cycle it back. And you will observe, this is called oxidation peak. This is a reduction peak. And the peak positions is dependent on the redox potential of ions. And each ions will have slightly different, sometimes very different potential. Just by looking at where the peak is, we can tell which ion is. And this amplitude of the peak is related to a concentration of the ions.
Yes.
So especially for the mobile sensor, it seems that you're using the audio
Right.
for the I/O bus. So, and also you had the battery on the other side. So is it because of the power consumption is going to be enormous compared to that?
We are necessarily need to use battery, but we use batteries because the voltage output from the audio jack sometimes not high enough to oxidize or reduce some ions. Okay.
So for the portability, you use additional battery to supply enough voltage for the sensor?
Well, for powering the sensor itself, we actually don't need the battery. The reason why we need the battery is because we need to raise the potential on the electrodes to some level. For example, 3 volts. That 3 volts output cannot be provided by the audio jack of the cell phone. Okay.
Then
do you still send the image data through the audio bus?
We don't have image data in this case.
Okay.
So this is our electronics data. This is our voltage and current data.
Okay.
Yeah.
So I assume you use nanohub to do all your analytical work, right?
Yes, of course, I hope so, especially for the SPR sensor case.